the processing is seaweed that is brushing up upon our shores and this crisis that we're having as a region, how well we can take this and create this into an opportunity. So this is a collaboration between the University of the West Indies in Mona and the University of Bahamas. And as I said, I would like to welcome you all here today. So first, I would like for the welcome and opening remarks from Dr. Kristen Welsh-Onwalo, who is the Executive Director and Assistant Professor of the Environmental Science Small Island Sustainability Research Complex from the University of the Bahamas.
Esteemed guests from the University of the West Indies, community partners, faculty, and staff, welcome and good afternoon. Thank you for joining us here today for this workshop on Sargassum, Opportunities for Commercialization. My name is Kristen Welsh-Onwalo, and I'm the Executive Director of the Small Island Sustainability Research Complex at University of the Bahamas. We're excited to partner with the University of the West Indies, Mona, for this workshop, and grateful that they share their expertise on this topic with all of us here today. Sargassum provides habitat, breeding grounds, and nursery areas and food sources for a variety of marine life, including fish, sea turtles, and marine birds. That's vital for the marine environment. However, warming oceans, changing ocean currents, and excess nutrients is causing massive sargassum blooms around the world. And with these blooms, sargassum washes up on shores and causes an aesthetic nuisance for beachgoers and tourists. Sargassum is a common issue across the Caribbean, and we're observing some of these massive wash-ups throughout the Bahamas. This issue requires a concerted effort across the region to provide greater understanding of the causes of these sargassum blooms, as well as the best sustainable management practices to address the issue. For this reason, we are excited to partner with the University of the West Indies to offer this workshop. This afternoon, you'll hear from researchers at the University of the Bahamas and the University of the West Indies on research efforts on sargassum across the Caribbean. You'll learn about the ecological implications of sargassum across the region, as well as the potential commercialization for it. You'll also hear about sustainable management efforts and the UE-led Sargassum Bioethanol project. Before we begin, I would like to thank Ms. Lanika Roden for organizing this event and the team of researchers at UE who are sharing their expertise with us here today. Without further ado, I'd like to pass this along. Is this a recorded talk next? Or back to Ms. Roden? Sorry?
It's back to Kelly.
Okay, okay. So without further ado, I'd like to introduce Dr. Kelly Kingan from University of the Bahamas and Assistant Professor of Marine Sciences, who will be presenting on some of her research efforts in Trinidad and Tobago.
Good afternoon, everyone. Second. Give me 1 second. Are you guys seeing my screen though?
Yes, we can see it.
Okay, excellent. All right. Welcome, everyone. Thank you for coming. So I'm going to speak to you for a few minutes about some of the potential opportunities for research and collaboration in the Bahamas, given some of the work that I've done in Trinidad and Tobago, or that I'm still doing in Trinidad and Tobago. So I'll just go ahead and jump right in. So as far as sargassum in the Bahamas, it doesn't seem to be as big of an issue as in some of the other islands, at least from my short experience has been being here so far. I've been here for about 1.5 years, so I could just be misinformed. This could also be due to a lack of good reporting of where there are influxes of sargassum coming in, and that might be somewhere we could look into trying to develop a better reporting system for the Bahamas. It seems like it rarely washes up in extreme amounts, not to say it's not here in presence, but it's usually in smaller amounts. And again, this is just from my experience on some of the islands and speaking to some of my colleagues. It may be worse at the southern islands of the Bahamas. as we don't get a whole lot of reports from down there. And we know in the Southern Caribbean, it is a very large problem, as well as to Jamaica and into the Gulf of Mexico. So it kind of seems like it's hits other islands before hitting the Bahamas really hard, but that's likely to change in the future. On several of our beaches, it is raked up or removed with tractors or other machinery on a regular basis for tourists because there are these small influxes that are very, very common. And I have seen it used as mulch on coastal plants, and some farmers and gardeners have reported using it for different agricultural purposes here, but I don't know of any commercialization of it in the Bahamas as yet. So here's just a map to show where the Bahamas is. It spreads over a wide expanse, and I think a lot of the sargassum that does reach our shores actually comes from the Sargasso Sea and not the sargassum belts from the equator that seems to be driving a lot of the issues across the other islands. But like I said, this could shift and change as we go through. I was recently in San Salvador, which is one of the Bahamian islands. I forgot to mention these stars here. So I'm currently in Grand Bahama, which is here. And we do get small little amounts of sargassum on the beach, not usually extensive amounts. Here in Nassau, I think similarly, they get small amounts where they have to rake it from in front of some of the resorts, but not kind of huge influxes. And then here's San Salvador, where I was just visiting for another project and came across quite a good amount washed up on the beaches, but nobody seemed to be complaining about it or having much issue with it. So I think it's more of a regular occurrence there. And you can see it's much more exposed to the Atlantic Ocean here. So moving on to what I've been doing with the sargassum in Trinidad and Tobago. It is a much bigger issue there. It washes up in huge amounts along mostly along the east coast of Trinidad and kind of the north, east and southeast side of Tobago, but it does come around to the other sides of the islands as well. And it can be quite massive influxes that cause big problems. So these impact coastal residents, the tourists, the fishers can't go out and fish. It's also going to impede any kind of recreational activity that happens along the coastline and much, much more. So these thick mats are common and greatly restrict all coastal activities when they are present. They're harmful to coral reefs, seagrass beds, mangroves, and other habitats because they block lights from getting to these areas. And as the sargassum starts to break down, there's a lot of toxins released, as well as it depletes the oxygen in the area. So it tends to have very negative impacts on these coastal habitats, and of course the fisheries and other things that we depend on when it starts to break down. So with my nonprofit organization called SHOPO, which stands for Conserve Habitats and Protect Organisms, we are working with youth, fishers, farmers, government agencies, and other nonprofits to develop innovative solutions for this sargassum issue. And the work we're doing right now is currently funded by the GEF program, the Global Environmental Facility, as well as is funded by UNDP, the United Nations Development Program. And for our project, we have 7 outputs that we're working on, although a couple of these are kind of shifting at the moment. But our goal is to educate 120 STEM students in sargassum biology, vessel logistics, and sustainable materials for a competition to design a device that the fishers can use to collect the sargassum. So we solicited STEM students to join this competition, and we had several training sessions with them, and then they submitted a design for their device. And then the top 3 contestants went on to actually build out their prototypes and trial them at sea. And then we had the top winner, who we're currently working on improving her device, and hopefully it will result in a patent and something that the fishers may be able to use in the upcoming months or years. Our outputs following that were to involve 125 mostly women and youth farmers across 5 farming communities in the use of Sargassum products and sustainable farming, as well as 5 women farmers provided with practical experience using and testing Sargassum agri products. And this is kind of slightly shifted due to the funders kind of wanting to us to change the direction of our product developments somewhat. So this might be not exactly happening in that manner. We're still kind of working out the details of that. We've already done our 5 fishers trained in the use of the Sargassum collection device through our trials with the contestants. We worked with local fishers to use these various devices on their vessels. We've collected nearly 36 van loads of Sargassum from the shores so far, just because our devices aren't currently collecting the Sargassum. There was some timing issues with when the devices were ready. The Sargassum kind of had already come in, so we were working on collecting the Sargassum from the coast while the influx was coming in. And we have the devices now, but haven't had much Sargassum since then. And then what we're really working on at this point is trying to develop 2 products from the Sargassum and market those and get them ready for sale and use. And I'll get into what those might be in a minute. But just wanted to show a few pictures from some of our students' activities. So these 2 pictures on the outsides are of the students, the top 3. These are 2 of the top 3 working in the workshop. We partnered with Massey Machinery in Trinidad, and they were able to use their professional workshop in order to build their prototypes out themselves. And this picture down here shows the 3 contestants with their little plaques, and some of our collaborators. We had various judges to judge their designs and how they functioned across different communities and nonprofits and government organizations, all working together to make this happen. And here, again, are the 3 contestants with some of their collection for the day. Unfortunately, there wasn't really much Sargassum for them to collect, so we improvised with some coconuts and dried Sargassum we had. And here's 2 of the designs by the 2 of the students here. One, he had kind of nets attached to these PVC poles, kind of like oars to more manually collect the Sargassum and then dump it in this net in the pirogue, just the local fishing boats. And then this design is kind of like a trawl with like 2 doors, a mini trawl to collect the surface Sargassum with those. And then our winning design was actually these barrels. It does need some modifications because it's quite heavy, but it works pretty well. The big blue barrels can be stored upright when not in use, and then easily released down into the water for collection. So this was our winning design that we're working with Navita now to try and make improvements and work on her hopeful patent for this. And as far as our potential product ideas, we initially were going to do something more agro related for the agricultural industry, but we've kind of shifted gears a little bit and are now exploring some products using the Sargassum to make paper with stationery as potential oil absorbent materials for for oil or gas spills, dyes for fabrics for the fashion industry or for textiles. So we're working with the fashion industry degree at the University of Trinidad and Tobago on that. We are also have been looking into animal feed additives and potential use for it as fish food or within fish food. And then some of our original agro product ideas were compost, fertilizers, and mulch, which we've tested in various ways already, at least started to with those products, and the others are more in the development phase. And of course, there's lots of opportunities for products, biodiesel and fuel and all kinds of things that you could potentially make sargassum from. So we're interested in trying to transfer these methods to the Bahamas, where we could do similar education type programs and incorporate our local fishers, farmers, government ministries, nonprofits, and community members here to try to alleviate this problem. So we've realized through this work that collaboration is key. Even though the issue at the moment might not seem that great in the Bahamas, it could definitely worsen in the near future. But by working together, we can prepare and be proactive in our efforts. We can start to experiment with the sargassum that is available here to develop products for local use and potentially even exports. The Bahamas could also be a potential place to store and process excess sargassum that's accumulating across the region. And the climate here is actually highly suitable for drying the sargassum, which is a typical step in the early processing of it. So we could potentially help with that side of the commercialization. At our large EEZ economic exclusive zone, most of the Bahamas is actually water, provides opportunities for future collection at sea. If we have some of these devices available, we can go out far from the islands and potentially find these huge amounts that may be an issue down the road. And with that, I'll thank you guys for your attention and happy to take any questions if there's time for that. Thank you.
Okay, so thank you so much for that, Dr. Kim. That was really insightful. And we love to hear what's happening, what you've been doing with the sargassum. So now we'll move on. So are there any questions, or maybe we could take some questions after? But we will move on to Professor Mona Weber, who isn't here in person with us today, but we do have her pre-recorded welcome and opening remarks from the University of the West Indies, Mona. And I will play that for you right now. She's the head of the Department of Life Sciences at the UW Mona, and also a colleague to the UW Sargassum Project. So.
Good day, colleagues. I am Mona Weber, head of the Department of Life Sciences at the University of the West Indies, Mona campus. It is my honor and pleasure to welcome you to the project workshop on advancing commercial scalable processing or storage of Caribbean sargassum. The sargassum inundation of the Caribbean has continued virtually unabated for the past 15 years and has therefore captured the attention of a wide cross-section of persons. We in academia started out seeking to understand the phenomenon, to figure out why the golden tides were coming, where they were coming from, and where they're likely to end up once they entered our coastal waters. After many years of research, we now accept that the inundations will continue, and we are now at this stage of increasingly seeking to valorize and use the algal biomass. Through our research, we know the dominant species and morphotypes that are contained in the pelagic floating sargassum. We now know the quantities that we're likely to receive and where sargassum is more likely to be found across our shores and in our coastal waters. We have also documented the deleterious effects on our coastal ecosystem, and the exploration of the constituents of the sargassum and the potential for value has led us to the stage of exploring not just valorization, but actual commercialization of these constituents. Life Sciences has been involved in several sargassum projects with local and international partners, but this project provides a new opportunity. We now would like to get into the process of realizing commercialization and to work with local stakeholders and partners to promote the use of sargassum as a biofuel and to take this beyond the demonstration scale. So we will and are involved in technical and economic feasibility studies, as well as evaluation of the current and potential demand for bioethanol in Jamaica. And this gives us the potential to have this introduced as a source of fuel singly or introduced as a mix with other traditional sources. And so this is new ground for us in Jamaica. And as we seek to share what we have learned so far with you, our Caribbean partners, we also would like to get your valued insights as to how we can realize the objectives of our project. And so we look forward to a fruitful day of exchange and interaction. And we're really grateful for your giving us your attention and your time. Thank you again, and thank you for allowing me to welcome you.
So that was Professor Mona Weber giving her welcome remarks from the University of the West Indies. And from that, I would like to invite Albert Turner, who is a climate and environmental advocate, who will speak on youth, climate, leadership, and research pathways. So we will continue to speak on that. Thank you. So good afternoon, everyone, and those who are virtual. First, I'd just like to thank the team from the University of the West Indies on the campus, and also the team here at the University of the Bahamas for hosting this workshop. I'm not going to take up too much of your time because I know we all want to learn about the opportunities and challenges that are surrounding sargassum in the region, but I was asked to speak about something connected to today's conversation when you think about the role of young people in shaping Caribbean environmental research and how early exposure can completely change the trajectory of a young scientist. And I'm sure Dr. Bethel could relate when we all think about our undergraduate years, and some of us were absolutely certain of our career direction, and others were just trying to figure things out along the way. I was firmly in the first group of people. I walked into the University of the Bahamas very confident that I thought marine science was my path, and I was on my way to becoming a marine biologist. And spoiler alert, I am in no way a marine biologist. But what did happen was something more meaningful. My journey into environmental research, and the thing that grounded me the most began right here in the halls of UB through the Small Island Sustainability Program. The program shapes my foundational love for research and field work.
And that came with my first real project. I had the opportunity to work on a project focused on understanding vulnerabilities, identifying opportunities for climate resilience on Grand Bahama post-Hurricane Dorian. And it was my first true field experience, and I owe all of that to Dr. Anwala. And it was the first time that I felt like I was contributing to work that mattered outside of classroom assignments. And being a part of research that eventually became published gave me a sense of responsibility and ownership. And it just showed me how powerful research can be when it's connected to home and when we reflect the needs and the stories of various communities that are navigating real challenges. And before that project, I really didn't think that I was ready to engage in research. I thought the idea of participating in research projects felt really daunting. However, when I stepped into the field, it had me realize that research isn't just about data and publishing a paper. It's about fostering community and curiosity, creativity, community resilience, and learning how to problem solve in real time. And this is why I believe that early exposure is formative, and that one opportunity in one project can shift someone's entire path. So I've been able to study here at the University of Bahamas and also at the University of the West Indies, Cape campus. And those experiences showed me just how rich and how diverse and how necessary Caribbean environmental research is. When we look at the different institutions and the different countries, they all add new layers of understanding to our evolving environments. They remind us that Caribbean research and science is strongest when it's collaborative, when our islands can learn from each other, when we share knowledge instead of working in silos. And I truly believe spaces like this workshop help build regional research networks, and that will shape the next generation of Caribbean science. And this dynamic that we are trying to actively build in the Caribbean, where emerging scientists and regional experts are leveraging our local knowledge and contextual expertise, help us to address our pressing challenges, able to contribute to global discourse. Because the truth is, if we don't really engage in research and encourage others around us to do so in our region, someone else will ultimately write our narrative for us and tell our story back to us. So it's imperative that our voices and our research ought to be leading not only in our journals, but outside the halls of academia as well. And I do think at UB there are so many opportunities to get involved when we think about undergraduate research, faculty-led projects, fieldwork, internships with non-governmental organizations. And across the region as well, UB is doing an amazing job at taking stock of the various challenges that we're experiencing and turning them into opportunities and offering opportunities to individuals to dive into different tracks that focus on Caribbean resilience strategies, which play a direct role in building a more sustainable future for our islands. And so to just wrap up, I just wanted to encourage those in the room and the students on the call to just take the first step when you consider research and consider your career path, because our region does need scientists who understand and who are able to question, who are willing to collaborate, and who will look at challenges and see room for opportunities to build a better Caribbean. Thank you.
Okay, so thank you so much for that comment. That was actually really great. And I think I could actually relate to that because I'm also an undergraduate. Well, not also, but I'm an undergraduate researcher. So it's really relatable, and it does, yeah, that's really, we appreciate you for that. So now I'll be playing a pre-recorded presentation from the lead investigator of the Sargassum Review Project, Ali Mona. So Dr. Chinapale Bhaskar, who is also a senior lecturer in the Department of Life Sciences, he will be giving an overview of the US Sargassum project, which I'll be sharing with all right now.
Good morning, esteemed colleagues, partners, friends, and fellow stewards of the Caribbean environment. It is a both pleasure and a privilege to welcome you to this crucial workshop. We are gathered here today, united by a common challenge that has been washing up on our shores for a decade, the Great Atlantic Belt. What began a crucial anomaly has become a persistent emergency. Our world famous beaches, the livelihood of tourism, are being inundated. Our mangroves and coral reefs are suffocating. Our fishing communities and industry are facing disruptive livelihoods, and public health concerns are rising from the decaying biomass. But today, we are not here just to describe the problem. We are here to explore a transformative opportunity. We are here to discuss how we can turn this environmental crisis into a beacon of innovation, a source of renewable energy, and catalyst for new blue economy in the region. Sargassum species, as we know, affecting us, dwell into unique biological composition and focus on one of the most promising avenues for valorization, that is what called biofuel production. I will also share exciting news about the Sargassum Bioethanol project, a significant grant award from the UK Foreign Commonwealth Development Organization Office, and our experiences on the ground so far. The project explores the potential of using Sargassum as a bioenergy source, specifically for the production of bioethanol, which can serve as an environmental friendly and cost-effective alternate to the fossil fuels. Besides this, we are also exploring the potential of use of Sargassum as a source of alginic acid for various applications in agriculture and industry. Their problem is key to success as an invasive species and their potential as a feedstock for rapid growth, high biomass. In case of rapid growth, they reproduce asexually through fragmentation, allowing for exponential growth fueled by nutrient-rich upwelling and possibly terrestrial runoff. While seeing the high biomass, they yield out double their biomass and in 20 days, leading to staggering volumes we see. We look into the biochemical makeup, which is a foundation of its biofuel production potential. Sargassum is one of the biorefinery which we are waiting for. Its composition primarily consists of polysaccharides, as we know, that is 35 to 45% dry weight, like alginates, phycoidins. Alginates are structurally polymers used for food and pharmaceutical industries. Phycoidins, sulfate polysaccharides, with a bioactive properties. Cellulases and hemicellulases are primary targets for the bioethanol production. These long chains of sugars can be broken down, that is hydrolyzed and fermented into a bioethanol production. Apart from this, proteins and minerals also play a major role of proteins of 10 to 15% potential for animal feed and fertilizer, minerals for around something like 20 to 35. Notably, it is a high arsenic and heavy metal content, which is our primary challenge and critical consideration for any use. This composition makes it a compelling, if complex third generation biofuel feedstock, avoiding food versus fuel debate associated with crops like corn and sugarcane. Our consortium has been awarded a major grant from UK government, that is Foreign Commonwealth Development Office, under commercial viability of Sargassum. The production of bioethanol from brown algae Sargassum using acid and enzymatic hydrolysis and its application for alginate synthesis from its cellulosic cell wall, initiating a design to move this technology from lab towards a commercial reality. The award is a testament to the FCDO belief in Caribbean innovation and its commitment to funding sustainable solutions to global environmental problems. The project has some following core objectives: selection and procurement brown algae that is Sargassum species from coastal areas of Jamaica, processed for better cellulosic salvo with different pretreatment methods, and proximate analysis were performed after processing the sargassum, estimate total carbohydrate, dietary fibers, lipids, reducing and non-reducing sugars and glucose, extraction of alginate from sargassum, also done an analysis of sugars and other soluble sugars done through HPLC for better understanding of the yield of ethanol in the final output. Total phenolic compounds and total soluble lignin and elements are estimated. By taking the sargassum and their pulp along with alginate were analyzed by FTIR analyzers and analysis. Saccharomyces and fermentation ethanol estimations are carried out by standard procedures using commercial strain of Saccharomyces cerevisiae and cellulose from Trichoderma ricci. So the fermentation of acid and enzymatic hydrolysis were carried out separately, and ethanol is estimated by gas chromatography. And these all are done. At present, we are at the statistical analysis of results to prepare the final draft and to submit by November 15th, 2025 to the FCDO UK International Development. Thank you one and all. There are some challenges to face regarding this work of delaying chemicals and other experimental instrument usage, but we are going to face a better challenge after completing this project to proceed further on this particular bioethanol biorefinery project. Thank you, one and all. Thank you.
All right, so that is Dr. Rahul, and he's the lead investigator on the project. He would have given you a little synopsis as to what the method was the method employed to actually work on the sargassum and extract the bioethanol. But to go through that with you in a step-by-step motion, we do have our Senior Technical Officer, who is Ms. Anne-Marie Hosang, who will join me on stage as she will give us an overview of the experimental procedure that was actually carried out by the team. All right, so while it is that we have the presentation being loaded, do we have any questions so far for the team?
Right. I think my first question is that in terms of the collection of the sargassum, that method that won the competition, it may be a bit problematic in terms of if there's a large concentration of sargassum, the boat won't be able to move forward because sargassum is at the bow, right? Get caught in the propeller. So why not have a let's say something at the bow of the boat to push sargassum around the hull of the boat itself, and then arms extended from each side of the boat to actually hold onto the sargassum as it moves forward. So you can now push it as you go.
Okay.
Because otherwise, like if you collect it in the buckets, you have to now be very strategic on how you're gonna get from place to place. But if you choose again, to put the structure around the boat and then arms extended out and put it's in the water that has claws on it, you can now push it, sargassum the way you want it to go. And if you were to automate it using automate a jet ski type or size or a skiff size device, you can run multiple of them going at the same time rather than one boat at a time with 10 different people.
Okay, that's a very good point. I'll say that for the most part, in terms of when we've been in the field and actually collecting the sargassum, since I would say a year and a half, the way that the sargassum has been traveling, it's more in small clumps, so you still have a lot, but that large mass that we've been used to probably 2 years, 3 years ago, that we've seen that mass size, as soon as it gets to the actual coastline, it actually breaks up, so it doesn't come
I see.
towards the coast in that large mass. So it doesn't impede the boats when the fishermen go out to catch it. Yeah, we don't have that challenge yet.
I see.
But we've been studying and watching the different parameters because we know what has been driving the promulgation of the bloom itself, which is, climate change and other factors such as nutrient runoff in the Amazon. And so we might see those large blooms return, but we have not. We would love to see it return actually, because now we're more prepared. The private sector is more engaged and they are looking eagerly for Sargassum to come up on their shores, because there are small businesses that have kickstarted their whole business model and concept on Sargassum. And I guess Ruth in her presentation will discuss the issues that we've been having with seasonality. And it has been very hard to predict when it will come, even though we have the monitoring system. But still, when you have a business that's depending on hoping to see some Sargassum wash up on the shore by next week, and we're not seeing any on the radar, it's a different conversation.
When you say on the radar, do you mean a literal radar or a metaphorical one?
Metaphorical.
Oh, the other suggestion was that if you are to be doing forecasting, the satellites are good, but then I catch the small ones that your boats can catch, right? It's too small. I would then suggest you to take fly a drone on a regular basis, and fit it with an automatic detection software, where you just make it one quick pass, right? It will detect where these things are, and then send out another AUV, a boat one, to go and collect that and bring it back, rather than relying on a large vessel that requires, of course, 2 people and fuel itself.
Yeah.
An electric power AUV would go out, scoop it up, bring it back, and this way you can now minimize having to be scouting on a regular basis, which would of course eat into how much, what is your return on investment? The more fuel you put into this, the less your ROI, the smaller your ROI would be.
And that's an excellent point, because one of the core objectives of this project is for the actual collection and processing to be economically viable.
You can automate the process. You can really automate it.
Yes.
Drones now, you can set, let's say if you have a given area, you can select a path and have it scan the entire thing, and then look at the ocean color, the difference in ocean color, because there will be a difference. And then from that, select out images that, you train an AI model saying, this is what Sargassum looks like on the ocean surface. This is how much that looks like. And then you go out, do the imagery, bring it back, process it, half an hour worth of work. And then you make a decision whether you're going to go out and send a boat, or what size boat you want to send out. So AUV is something small, but if something much larger, then you take the large skiff with a modified collection system that has the arms, they extend out, claw into the water, and then drag all that to the collection area. It reduces the amount of manpower you need. And once you reduce the manpower,
it's actually very useful. We'll definitely capture this for our reports. I think this will feed into phase 2 of the project as well, which we would invite you to be a part.
Yeah,
because you have a lot of knowledge and innovative ideas, because we've been in airplanes and when we fly over the coast of Dumbrook, you can see the Sargassum. It's very visible because it's bright yellow. And in the open sea, you can see that it's in larger mats, but it's just when it comes to the coast, it's broken up and fragmented. And so that makes it, you're just catching
the bits. So this
bits and bits. But I guess if you were to travel out in the open sea and catch it using the method that you propose, which I absolutely like, I guess it would be up to now testing the feasibility of.
Well, I think your biggest hurdle would be you need to build a drone big enough that can loiter in the atmosphere for, let's say, several hours, not something for half an hour. So you want to have something that is like a propeller, right? But have inside the wings itself bladders that can take hydrogen. So it will be light anyway and reduce the amount of energy you need to actually stay aloft. And you have it just circling around the area over and over, just taking images. Relay those images to a station, have them processed automatically, and have a human to check them afterwards. And then you say, okay, well, the biomass has reached a certain level where it's now economical for us to go out and we collect. We're not collecting 1, 2, 3 bits. No, we're going out to collect, tons of it all at once. And then you store that and you spread it over time to eliminate the issue of seasonality that she will have.
Yes.
And if you wanted to monitor your seasonality, and it is dependent on ocean conditions around the Amazon at the mouth of the river, you want to talk to the Brazilians who are monitoring that system very carefully and have them send data to you.
Oh.
And anyway, please continue.
We will talk. So I will now hand over to Ms. Anne-Marie Hosan. There's another question in the chat.
Oh, I can't read on my screen.
Oh no, he's going to bring it to us. Okay.
What is expected to be done with the leftover biomass once reverse is extracted?
Excellent question. So you're preempting us, but we will get to that in terms of the different applications of the actual residual biomass that would be left after we process the sargassum. But just to give you some things quickly off the top of my head, it can be used as a fertilizer in agriculture, depending on the concentration of potassium and magnesium. But there are different scenarios to that. So if it's high yielding, then it can be used as a fertilizer, as a biofertilizer. If we find that potassium and magnesium, depending on the method that you use, and the biomass left over is low in potassium and magnesium, then it can actually be used as a alternative for, I think, as a growth medium that we can feed the necessary nutrients in. So there are different applications that we'll get to in the different presentations. So I'll hand over to Ms. Hosan for her to walk us through the methodology that would have been employed in this Sargassum commercialization project. Ms. Hosan has years of experience as a senior technical officer at the University of the West Indies. Before that, she would have worked at the Bureau of Standards Jamaica. So when you talk about a seasoned scientist who has joined our project as technical lead, and she will share with you on the methodology for bioethanol and bioproducts extraction from the Sargassum species. So please pay keen attention, because our goal here today is to share everything with you. So if you want to literally take the blueprint and redo the method yourselves, you can.
Good afternoon, everyone. When we hear the word sargassum, it's something that is a nuisance to us, and we have challenges not only in the Caribbean but in the wider region. It even goes as far as West Africa. Hence, we are here today to seek the pathway or to discuss pathways that can transform this nuisance into a resource. And before I delve in my presentation, I'd like to introduce myself. I am Anne-Marie Hosan, a scientific officer by job description from the Department of Life Sciences in the Faculty of Science and Technology. My role in this project is I am one of the technical lead, and the topic of my presentation is value-added potential, assessing sargassum viability for bioethanol production, concentrating on the comprehensive methodology for bioethanol and bioproduct extractions from sargassum species. Excuse me. Well, my outline for today, I'll be giving you a brief introduction and aim. I'll be showing you a summary of our workflow chart. I'll be speaking on different phases such as sargassum pretreatment, the proximate composition analysis, saccharification, otherwise known as acid hydrolysis, fermentation to bioethanol, and alginate co-product extraction. Sargassum species influxes have been washing up on our shores of the Caribbean islands and the Gulf of Mexico at a record-breaking rate, creating ecological, economical, and social issues for a variety of countries within the region. This led to decision to investigate and explore what benefits, if any, can be obtained from this macroalgae. Investigation have shown that the seaweed have been used as a generation feedstock due to the, One minute, please. Sorry. Sorry, let me read over that slide. Investigation have shown that the seaweed have been used as a generation feedstock due to its high growth rate, no arable land or freshwater needed, and no lignin in its cell walls. This macroalgae has an abundant reservoir of untapped potential of sugars and lipids, and the biomass is currently a widely used source of renewable energy. Its utilization is steadily increasing due to growing concern about the adverse effect of fossil fuel consumption and the detrimental impact on human and the environment. Hence, the investigation of sargassum and its commercial usefulness as an additional source of biomass for bioethanol production. The aim of this research is to investigate the potential of sargassum biomass as a sustainable, renewable source of bioethanol production. The specific objectives are to determine the approximate biochemical and physical composition of sargassum, to break down complex carbohydrate, example alginate, laminarin, into simple fermentable sugars, to convert the fermentable sugars in sargassum, hydrolysate, to bioethanol using Saccharomyces cerevisiae, to extract sodium alginate from hair-dry sargassum. The project outcome, we have 5 outputs. First is the morphological characterization and procurement plan. The second is the pretreatment and proximate analysis. The third is the acid hydrolysis and fermentation. The fourth is the stakeholder engagement and findings. And the final one is the final report dissemination. But today I'll be focusing on output 2 and output 3. Pretreatment and proximate analysis. This evaluates different pretreatment methods for better yield of carbohydrate. Characterize proximate analysis such as moisture content, volatile constitutes, ash content of selected brown algae, sargassum. The proximate analysis will follow after processing the sargassum and estimation of total carbohydrate, lipids, reducing and non-reducing sugars, and glucose will be done. Estimation of reducing sugars for acid hydrolysis after pretreatment. The acid hydrolysis fermentation. This is to convert sugar to ethanol using fermentation process. Estimate ethanol by physical, chemical, and biological methods from the biomass of Sargassum. Saccharification, otherwise known as acid hydrolysis, fermentation, and ethanol estimation will be carried out as per standard procedures using commercial strain of Saccharomyces cerevisiae. Estimation of ethanol content using gas chromatography and high-performance liquid chromatography, GC and HPLC method. Extraction of alginate from Sargassum as per Zuhra et al. In 2008 using formaldehyde alkaline treatment method. Characterize the extracted alginate for its properties and applications. Analysis of Sargassum and their pulp along with agar and alginate standard as well as extracted by the FTIR analyzer. This is the workout schedule that was done. Phase A was the Sargassum harvesting. Phase 2, showing the pre-treatment method. Phase 1, showing the Sargassum pre-treatment. Phase 2, showing the proximate composition analysis. Phase 3 is the saccharification or acid hydrolysis. Phase 4, fermentation to bioethanol. Phase 5, alginate co-product extraction. So phase 1, Sargassum pre-treatment. The objective of this is to prepare raw Sargassum for downstream processing. The methodology is for the collection and washing. The Sargassum was collected offshore from Port Royal Keys. These samples are a mixture of different species, which are the Sargassum natant and the Sargassum fruitant. The Sargassum was rinsed with tap water to remove excess salt, sand, and epiphytes. This was then dried at room temperature with a standing fan for 1 week. After it was dried, it was grounded into fine homogeneous powder using an industrial blender. It was then stored in airtight containers in a refrigerator. Here on the right side of your screen, you can see the air drying process going on. And at the bottom, you can see the instrument used for milling or grinding. Phase 2, which is approximate composition analysis. And this is to determine the approximate biochemical and physical composition of Sargassum species. Here we have done the moisture content, which is the change in mass after drying at 105 degrees for 6 hours until constant weight was reached. We have the ash content, where residual weight after incineration in a muffle furnace at 550 degrees Celsius. We had the crude protein done by the Lorry method, and we have the crude fat done by the succinate extraction, and total carbohydrate by difference, which is calculated at 100% minus percentage moisture plus percentage ash plus protein plus percentage lipid. Here, this picture is showing you, we have 3 of them. Picture A, picture B, and picture C. Where picture A is showing moisture samples in the glass petri dish. Picture D is showing samples, the ash samples in crucibles, and for C, it is showing the moisture samples in a desiccator after drying, and this is to prevent reabsorption of moisture. The saccharification or acid hydrolysis. The importance of this is to break complex carbohydrates into simple fermentable sugars. Here in acid hydrolysis is one of the limiting steps in the entire process. The production of ethanol is very dependent on this step for breaking complex bond in the polysaccharide chain into simple ones. They are governed by different parameters such as acid type, acid concentration, reaction temperature, biomass to liquid ratio, and reaction time to optimize for extraction and release of the high amount of sugars. The methodology that we use in this process was adopted from a paper produced by Wardiani and Ariani in 2019. This method was modified and was made to include the use of different types of acid at different concentration over different incubation time and at different temperature to determine if there is a correlation between these factors and greater yield of Sargassum sugars. So these are the acid used. We use hydrochloric acid, sulfuric acid, and nitric acid. Water was used as a control. And the parameters we use, the acid concentration we use was 0.1 mole, 0.5 mole, and 1 molar. And the varying temperatures were 60, 80, and 100 degrees Celsius. The reaction time was 30, 60, and 90 minutes. The solid to liquid ratio is 3 grams in 200 mils of the various acid concentration. And it's important to note that all trials were done in triplicates to reduce experimental errors. For the acid hydrolysis, the procedure is that, as from the previous slide, the Sargassum powder was mixed with the requisite acid solution in covered vessels. It was heated at varying temperatures in a water bath for a specified reaction time. The residual C biomass was then cooled, filtered to separate the filtrate from the solid residue, and samples of the hydrolysate removed for a later proximate analysis. The samples of the proximate analysis were stored at -18 degrees. The remaining hydrosylates were neutralized at a pH 7 using 20% sodium hydroxide and potassium hydroxide. It was autoclaved at 15 psi at 101 degrees Celsius for 15 minutes. In order for us to know whether the Sargassum can be used to produce bioethanol, there must be simple sugar present. So we went ahead to do some confirmatory tests to see if there were any simple sugars present. And the first one we did was we used Benedict's test for reducing sugars, which we used Phelan's A and B solution. Here we had 1-to-1 ratio of Phelan's A and Phelan's B solution were placed in a test tube, and 1 ml of the test sample were added and mixed. The sample were placed in a 60-degree water bath for 10 minutes, and the color change was observed, which we can see on the pictures on the right side. The color change was from the blue to brick red. So A is showing the reagent, and B is showing a positive test for reducing the presence of reducing sugars. We also use the dichromate redox test, where we use 1 ml of the test solution was added to 2 ml acidified dichromate solution, and the mixture was placed in a 60-degree water bath for 10 minutes. And as you can see on the right side, the first one, A, you see an orange coloration, which that's showing that there is no simple sugar. But after adding the test solution, we had a color change, as you can see in slide B, where it changes to green, which indicate the presence of silver sugar. Now, further analysis of the hydrosylates will be analyzed for total reducing sugar content using the HPLC method, and we also can use the modified 3,5-dinitrosalicylic acid method using glucose as a standard. Phase 3, fermentation to bioethanol. The objective is to convert the fermentable sugar in Sargassum hydrosylate to bioethanol using Saccharomyces cerevisiae. Now, it's important that we use a competent microorganism to convert various fermentable sugar to ethanol, example, the cerevisiae, Saccharomyces cerevisiae, otherwise known as the baker's yeast. First of all, before we can use the yeast, we have to prep it. We have to give it some nutrients, some growth nutrients, which we use the YPD, which is the yeast extract peptone dextrose medium, which is a non-selective nutrient growth medium. And here we would put the yeast in the broth for 24 hours so that it can get some energy to do its work. And now the fermentation medium would be the hydrosylate from the Sargassum filtering. So in detail, we inoculated 0.5 grams of the Saccharomyces into 200 mils of the YPDA medium for 24 hours at 30 degrees and was shaken at 90 revolutions per minute. After 15 mils of the yeast was placed in 150 mils of the hydrosylate. That is a 10 volume by volume inoculum to hydrosylate. Incubation was done anaerobically at 30 degrees for varying incubation times, such as 72, 84, and 96 hours, respectively. Let me go back. I can show you. You can see on the picture the setup for the fermentation process. So after fermentation, the samples were filtered to remove the yeast cells. So the filtrate was now analyzed for the ethanol content, and this was done using GC and HPLC method. The sugar consumption monitored by measuring residual sugars to calculate fermentation efficiency. Phase 5, alginate co-product extraction. And the objective is to extract sodium alginate from hair-dry Sargassum. The procedure is that we added 25 grams of the dry Sargassum, and it was treated with 2% formaldehyde for 24 hours. It was then washed with distilled water. The Sargassum was then treated with 0.2 molar hydrochloric acid and shaken for another 24 hours to convert the alginate to alginic acid. There was a second wash with distilled water. After the washing, the Sargassum was treated with an alkaline solution, which is 2% sodium carbonate, to solubilize sodium alginate. For precipitation, the alginate solution was filtered and the alginate precipitated by adding 95% ethanol and acetone to the filtrate. The recovery of the precipitate was collected and dried and weighed. This is pictures of the process of extracting alginate. Here in slide 8, ethanol acetone precipitation of sodium alginate. Here in B, we are showing where it is your drying of the sodium alginate precipitate, and for C, is showing the Sargassum residual forms. Lessons learned. First, you have to start off with a good sample. Samples that are fresh, non-degraded, and contaminant-free. Example, no other seaweed or debris will mix in with the sample. This reduces pre-treatment time. No need to do additional sorting to remove contaminants or contend with dumping or degrading materials. Degraded materials reduce optimal yield for simple sugars, which in turn reduce bioethanol production. Establish protocol and system for quick pre-treatment and downstream processing. Sargassum degrades quickly. Losses can be reduced if protocols are executed in a timely fashion. Reduce waste and ensure good yield in high quality production. Bulk sargassum is characterized by its high salinity. Therefore, it has to be washed well in order to remove excess salt. Optimizes the sugar release, the carbon source for yeast during acid hydrolysis. Excess salt results in high salt stress condition for yeast during fermentation, which reduces ethanol production. Key advantages of the process. The feedstock uses abundant, non-food, non-lignin-free biomass. Method employs a specific, efficient, and environmentally friendly process. The dual impact contributes to renewable energy goal while mitigating a critical ecological issue. These are the different references that were used. Turning sargassum from a problem into a resource. Thank you.
Oh, it's just me. So it's basically just me. So I have a question for you.
Oh, you have a question?
The report was very detailed. I think my only question is in terms of the amount of water that is used to desalinate the sargassum. The concern, of course, is that when you do this on an industrial scale, a vast quantity of water will have to be used. That's going to take away from other usages of that water. But also when you dump that water, where are you going to dump that to? So has that been considered within the entire scheme?
No, we haven't. We just wash in
I get that, but
yeah.
is that, like,
but we don't.
It's something that we would take into
Yes,
consideration
yes.
for the final report, because that's where we'll be doing the models in terms of looking at the commercial viability. So we're currently in the process of doing exactly that. So I think that's a very important point for us to note in terms of where that vast amount of water will come from, because it's very important to watch the sargassum because it cannot be used with the salt and the electrolytes that's on it.
do you have a salt industry in Jamaica?
A salt industry?
Yes.
We do. There is a factory that produces salt.
Right, and why not have the production of sargassum be paired with that? So once you finish with the fresh water and the salty water now, you send it off to the salt plant where they can use it directly.
Okay, that sounds good in terms of.
It eliminates the pollution issue. If you're sending it over into the salt production facility, because they will use that and then sell it off. And you can now use that to reduce the overall cost of doing this, because now you have another product to sell.
Yes.
So the wastewater, the highly saline water is not a waste product. It is a product unto itself if you are positioned to send it to where it needs to go.
Yes. Thank you. Any other question?
Okay, thank you.
I have a question here online.
Hello?
Yeah, so I understand that you said that you use tap water to wash the samples. Could you have used deionized water instead of tap water? And the reason I'm asking is that with tap water, you have suspended impurities that can actually be in these tap waters. And so by washing your samples with tap water, although you're getting rid of the impurities that are already in the sample, you're adding back impurities that can be part of the tap water. And so that in itself would then introduce a confounding effect in your models, sorry, not your modeling, your statistical analysis. And so that can introduce the impurities, and so by introducing those impurities, that can be part of the analysis in terms of what you're looking at in terms of extract, etc. And I think you also mentioned that salinity in itself can have an impact on the fermentation of the yeast. And then also with tap water have some level of salinity depending on where the source of the tap water is actually coming from, or if following event, for example, following events of major hurricanes or even hurricanes in itself. So why use tap water instead of deionized water?
We use tap water because it was readily available, but we can think about using deionized water next time.
Okay, thank you.
We do consider that the tap water will have ions in it, but I don't think it would necessarily be a major concern. But as you said, we will think of using the deionized water.
It might have a major concern because we were talking about biogeochemical processes, and some molecules themselves are very unstable, and so just the least reaction with some impurity can just basically change the entire results. You may not be able to see it with the naked eye, but then there's going to be some kind of reaction going on, maybe at a micro scale level, that may have an impact on the actual inaccuracy of reporting the results. So
Yes.
we probably want to take into account.
Yes, we will take that into account.
If you want to take two experiments, one with the deionized water, one with tap water, on those two, and see what the difference is. Now, if there's a measurable difference in terms of using deionized water, then you need to put the extra effort to get it, right? Because it may be worth it. But until you quantify it, you can't really know if it's, as you said, I'm not a chemist, so I will bend to what he has to say. But the reality is that when you use the tap water contains whatever is in the tap water. And if you don't know, right, if you don't know, when you're looking at results, how can you know if you're seeing the results from your experiment in terms of this is what the sugars are versus what is coming up from the tap water itself? You cannot know unless you do that experiment.
So take, for example, like on Grand Bahama, prior to the major hurricanes of 2004, and especially one that recently happened in 2019, the water that usually comes from the faucet have a higher salinity concentration than what it had prior to those major hurricanes. And so that in itself, where you have these major shift in the actual salinity or electrical conductivity concentration of tap water before major hurricane versus after major, I think it changed the water itself coming from the tap significantly. So that can definitely have an impact on any study, especially when you're dealing with, even with chemistry in itself, whenever you're doing like chemical analysis, you need to use deionized water. They don't use tap water to do any form of chemical analysis in lab. So they always use deionized water. But the dual study approach to compare to see if there's any significant difference, that could work fine to see whether you need to upscale using the deionized water versus tap water.
Okay, we'll definitely take that into consideration. Thank you.
You're welcome.
I have a question here online, if I may.
Yes,
My question regards the extraction of sodium alginate. How does that compare to commercial procedures for the extraction? And what does it look like when you all do the extraction of sodium alginate? Do you get a large yield from the Sargassum program, or is it low in your experience?
Well, so far we did a preliminary test, which we haven't really gotten into getting the figures. Since we have been here, we haven't done we haven't gone through the weighing and stuff of it. So we are yet to find out when we get back, when we are finishing up. Yes.
I see. Thank you.
All right.
I think that will give rise to the efficiency of the entire process. So for how many grams of seaweed do you need for, let's say for the sugars, how many do you actually need?
Yeah, we haven't gone through finishing, but yeah.
We ended up having to kickstart the stakeholder engagement process right in the middle of the experimental phase. So even now while we're here, there's someone in the lab doing some work. And when we get back, the next day we'll be back in the lab, but we finish everything on Friday this week. So.
Any other question? Okay, thank you.
Great, thank you so much for your well thought out questions. We really do appreciate it, and it will feed, all of these questions will actually feed into our final report. I can say that we'll probably consider actually running small experiments with the deionized water as a control sample and compare to see if the results that we get is alarmingly variable. At this time, I would like to invite to the podium Miss Ruthann Vassell. She is our experimental design lead. She will discuss the results that we've gotten so far, the seasonality and potential industrial challenges that come with sargassum. And so thank you so much for that introduction, Nika, because I actually forgot to introduce myself at the start. But as I said, I am Ruthann. I'm Miss Ruthann Vassell, and I will go into, I will just bring up my presentation real quickly. Okay, so all right, so I will go into a few of our findings, and I will also discuss the seasonality and potential industry challenges that we may face while working with sargassum in this commercial manner. So, as I said, a little overview. I will start with a little introduction, going through our findings. From our findings, or expected outcomes as well. When we go and do our analysis, I'll do a short discussion on the seasonality of sargassum and concerns that it may bring, and then I'll go into industry challenges, what we may face, certain hiccups that we may face while trying to commercialize this. So as we know, we have this issue at hand, even though I've been carrying read and through in the Bahamas, it's not really that large of an influx, but even as in Dr. King's presentation, they were saying there's a lot of this in Trinidad, and there's also a whole lot of this in Jamaica as well. Our beaches are getting taken over by this. It's really affecting even things like tourism and such. So we have this issue at hand. This is actually a picture that I took from Nepa in Jamaica, where it's a beach in Clarendon, Jamaica, where this sargassum is just completely covering the beach. You can't even see the water, you can't walk. Sargassum is covering it. So from that, in this project, we're trying to determine how could we use the sargassum, if it can be used for something else, how can we take this problem and turn it into an opportunity. So before we started doing our bioethanol conversions, we had to first test sargassum itself and see what is in the sargassum, what chemicals, compounds are in the sargassum. And I have a table here with a bunch of chemicals. We found out a lot more, but we took these out because they're fairly high concentration. I'm going to go through all of these, but from these, I have extracted some of them. So our preliminary testing revealed that we have these in significant quantities. So we have tetracosane and pentacosane. Around 15 to 20%, which are both long-chain hydrocarbons that are suitable in use of phase change materials or PCMs, which can be or are increasingly used for thermal energy storage in renewable energy systems. We also found octadecane, which is commonly used, a compound that's commonly used in cosmetics and pharmaceuticals for its emollient control release properties. So as Benita was saying earlier, when we were talking about sargassum, we're thinking of how it could be applied in cosmetics and so on. So a compound like octadecane makes it possible to be used in this in these industries. We have icosane as well. So icosane has a role in bio-based paraffin wax production and potential use in thermal energy storage. And it also has applications in energy efficient building materials, which also aligns with the project's goal to explore sustainable solutions, because we don't only want to see if bioethanol can be produced from it, but we were trying to see from different angles how this can be sustainable. We also have hexacosane. So given hexacosane's thermal properties, it has applications in lubricants and phase change materials, just like tetracosane and pentacosane, and can also be used in the cosmetic industry. We have from that now, okay. So from that, we have seen that we have these sugars in the sargassum, because of course we have to see there's sugar in there before we even start our fermentation, before we even start seeing this bioethanol that can be created from this. And we saw that there's glucose oxime, desorbitol, and ribitol as well. Which from this we concluded that yes, we can go ahead and try and create bioethanol from the sargassum. So as said in Ms. Hosan's presentation, where she was going through the method, we did our confirmatory test to roughly identify the presence of these compounds before we started doing our HPLC or gas chromatography methods or DNS methods as well. So I won't necessarily have to go back into this, but just to refresh, we did these tests on both the fermented samples and the hydrolysates. So when we did a potassium dichromate test, a redox test to see if these have reducing sugars, we had a before picture where before we heated it, we just added the potassium dichromate, and before we heated it, it's orange, and then it turned to this greenish brown color. Well, it'll be brown because greenish brown because the hydrolysate is off color already. So it turns to this, and then that which means that we got a positive test to indicate that there are reducing sugars, possibly reducing sugars that are present in both the fermented sample and the hydrolysate sample. We did this also as a control. So when doing the experiments, we did the acid hydrolysis, we tried to do the hydrolysis with water as well as a control to see if it's really the acid that's acting on if the actual sugars in there if something else. When we did this test with water, it remained negative, which is theoretically expected because we wouldn't expect reducing sugars to be in this mixture. We did the failings test to determine if there's a presence of reducing sugars. When we did this, we had a blue color from before, a blue solution from before, after heating it in the water bath. We had this red precipitate that's forming at the bottom, which is also an indication that we have reducing sugars present in our sample, which will also further indicate that we would have bioethanol if we ferment the sample. So we have other observations. Here where there is a slight alcohol scent from the fermentation mixtures. This is before we actually started doing our analysis on them. We're just noting what was there. So we had a slight alcohol scent from the fermentation mixtures. As the concentration of the acids and the time and the temperatures increased, so as these parameters got more intense, the hydrolysate mixtures are darker in color. except for, well, the exception of nitric acid, it actually got lighter, which is actually really interesting, but it got lighter, and upon concentrating these mixtures from the acid hydrolysis, because before we fermented it, we combined all of them and boiled up the water to concentrate or to get the most sugar from the solution before we fermented it. And we realized that we had this sugar, this precipitate that was formed, and that was hard to go back into solution, as more water left, which we can look at it and we just call it the seaweed sugar. So we have pictures right here where we have our sulfuric acid hydrolysis, the 0.1 molar at 80 degrees versus the 100 degrees Celsius. You can see the 100 degrees Celsius is darker in color and looks more intense. We have the nitric acid hydrolysis. You can see that these mixtures actually look yellow. So when our technical lead actually came into the lab, she was wondering, did you put sargassum in there? Because this doesn't look, it doesn't really look. But yes, we did. So that was also an observation. And then we have the seaweed sugar on the far furthest right in there, which was actually pretty interesting to see, truly. And we were actually thinking of taking it and drying it and probably performing an FTR to see what exactly is in that seaweed sugar. So from these, while we're doing our actual analysis, chemical analysis, we expect these. Also, theoretically as well, we expect that the sulfuric acid hydrolysis trials will generally have the highest yields of reducing sugar and bioethanol, and that nitric acid hydrolysis trials will have generally the lowest yields of reducing sugar and bioethanol. We also expect that as the acid concentration increases, as I mentioned before, the reducing sugar and bioethanol yield will increase, same with the time and same with the temperature. Or at least for those that we've done so far. So we see the 30 minutes, 60 minutes, 90 minutes, and with the 60 degrees Celsius and 80 degrees Celsius, 100 degrees Celsius. For those temperatures, because it could be a possibility that as those parameters increase, it could decrease, but for those specifically, we're expecting that they will increase. So we have our Sargassum seasonality. So with our other stakeholder engagements with our secondary research, we have found about it and said that Sargassum is a seasonal event. So with us commercializing this, it would be a concern that would we always have a constant flow of sargassum, a constant influx of sargassum to be used as feedstock to be used in industry. So sargassum season is usually peak in April and October. Seasonal growth is determined or influenced by many factors. So riverine nutrient discharges during the wet seasons, which during this time where we have increased nutrient discharge from the Amazon and Orinoco rivers, the warmer sea surface temperatures due to climate change. So due to climate change, these increased temperatures will actually help increase photosynthesis, which will increase the production and influx of the sargassum. So that will go into the increased sunlight, warmer temperatures, promoting rapid photosynthesis. And the longer daylight hours as well during these seasons. We have longer daylight hours which will provide these conditions. And then we also have the ocean current dynamics, which is transporting the biomass from the equatorial Atlantic westward to the Caribbean and the Gulf of Mexico. So these ocean currents during these times are actually bringing the sargassum into the region. So these are all factors that we all have to consider and try to predict as to how we can actually push this into industry. And this will also lead to the potential industrial challenges where this seasonality creates significant challenges. We have the irregular supply. So sargassum influx may not be straight throughout the year. Even if it's throughout the year, you may have larger supply than the other months. And this is also a challenge and concern. We have the variable quality. So quality in the very the qualities vary in the sense of it decomposing. Instead of it decomposing, which times of the year it comes and how long it may stay fresh, because when we're doing it, we don't really want it to be decomposing when we collect it. So we have that quality variance as well. Processing constraints, you need to know how to store the sargassum properly, how to collect it, when to collect it, and the conditions to have the sargassum at, which will go into the logistical and environmental issues. So even with the transportation of the sargassum and keeping it at the optimal conditions, you also have environmental issues in the sense where it will start to decompose. And when it starts to decompose, it may release, well, it will release these gases, toxic gases like ammonia and hydrogen sulfide, which contribute to respiratory illnesses and such. And there's also a concern where people are probably looking at sargassum and saying, this is just seaweed. There's nothing to be done with this. This is just garbage, it's just waste. So why would we want to waste our money, waste our time, waste our resources to use this for something else? So there's also those social factors that play in that as well that could impact industry in a sense where they're trying to engage with their stakeholders, and as I said, they say, no, we're not going to invest in this because it's seaweed. Why would we do that? So those are also challenges. But with all of that said, there is this call to action where it is possible and it is an opportunity. So instead of seeing the seaweed as just a problem, we can turn this environmental challenge into a model of sustainable innovation where it can be used in our energy sector. And that is the end of my presentation. So, does anyone have any questions?
You mentioned the first compound, sour tea. Right. You mentioned that it has some applications in renewable energy applications. Can you go into a bit more detail, please?
Into the renewable
yes. So you can store optimal energy, you say?
Okay, so I don't know fully the whole physics obviously of the compound, but it is used in heat. Well, it can be used in increasing thermal heat storage.
Let's say with molten salts?
Pardon?
With molten salts?
Yeah. So it can be used to increase thermal heat storage, which can be used in material science. So that's how it can be used in renewable energy systems.
See. And then with regards to your ocean dynamics, you do have a few currents that are flowing up towards the Caribbean Sea, but also you have like mesoscale eddies, a lot of eddies in the water as well. Would it be possible that when you detect the masses of sargassum out to sea, you fly a drone over, drop a buoy, a GPS buoy to track it as it goes? Is that being looked at as well?
To track it as it goes, as the influx goes up.
So okay, so you put
The movement of the
right.
mass.
So okay, so the satellite now will have a daily resolution, and so within a day, if it goes too fast, you won't see it at all, right? But if you throw the buoy in there, you get a reading every 10 minutes if you want. So you can really keep a track onto it as to where it's going, so that your issue of seasonality becomes less of an issue because you're not able to predict ahead of time where is it going to be, and because you have that drone, you know where it is and how much is it, and you can plan correspondingly. So would that be incorporated into, let's say, to industrialize the process?
Yes, I think that would actually help with it a lot, which I think is really why we probably need a physicist or two on our research.
Y'all don't talk to us.
But yeah, so for that to be a way of predicting seasonality, that would definitely help to push it more into industry and commercialization.
Yeah. So you can't control how much is going to be produced, but you can know when and how much will be there, and then plan accordingly.
Yes, and to see how much we can produce on what is there from what we planned and how to alter from there. Yes. Yeah. So thank you. So while we are waiting I will now invite Lenika, Mrs. Lenika Roden-Scott to give her presentation on the, or we have to.
No, I'll go first.
Okay, so we have Lenika going to do her presentation on the general opportunities.
Okay, so hi everyone, hi again. So I'll be talking about the actual opportunity as it regards to Sargassum. I realized that Ruth actually touched on quite a bit what was in my presentation. You'll see that I actually have the same table, the same results table. So that means that we'll breeze through my presentation pretty quickly. So we understand that it's a regional crisis. I don't need to go into the background, but I just want to say that with the Sargassum issue comes other issues. When you think about the problem of microplastics, I'll tell you a story. There's a beach in Jamaica that some Sargassum actually washed up on the shores. We're doing a collection in Portmore, and we saw a plastic product from Trinidad, manufactured in Trinidad. It doesn't sell in Jamaica, so that means it's literally that bunch of Sargassum is coming all the way down from Trinidad up to Jamaica. So it's contributing to the problem that we have in terms of plastic gathering up on our shores or washing up on our shores, and the whole issue of microplastics. And then when you look at the explosive growth rate of Sargassum, that's the other issue. So yes, we have seasonality, but at the end of the day, we do have quite a lot of Sargassum, a lot of it. The picture that Ruth showed you, I wish it was even a fraction of the problem. I know we probably don't have it as much in the Bahamas, but in Jamaica, the Dominican Republic, in Antigua, in Trinidad, there are quite a lot of the Caribbean countries that are dealing with this problem. It has cost us millions of dollars in terms of our tourism industry. It's very expensive to clean and to remove from the coast, because even with instructions and guidance from the National Environment and Planning Agency, you cannot just take a rake and try to rake the Sargassum off the shore, because you're dealing with a very fragile marine environment. There are small species to take into consideration, and you don't want to disturb that ecosystem. So that is another thing for us to consider. One of the things that we definitely aim to do is to optimize the conversion process. And so you would have heard Ms. Hosan mention the fact that we manipulated the acid types that we use. So we use nitric acid, we use sulfuric acid, we manipulated the different concentration of acid, we changed the temperature around, we changed the reaction time around, all so that we can find what is the most efficient pathway for sugar release and subsequent alcohol production. So really, why we decided to change the concentration, and in terms of what we found so far, connected with what Ruth would have presented on, is that when we increase the acid concentration, it proportionally increased both reducing sugar liberation and ultimately bioethanol production. When we elevated the temperatures, we're expecting that it should accelerate cellulose breakdown and improve overall bioethanol yield efficiency. But as Ruth would have cautioned, something for us to consider is the temperatures that would have been outside of the range that we didn't necessarily explore might not necessarily mean that it would optimize the reaction products or the byproducts in terms of getting a higher yield, because it might actually affect the plant material and cause more damage than harm. And so at the end of the day, while we continue to work on this project, and as we mentioned, there is a phase 2, we aim to deepen our understanding of Sargassum's potential as a renewable energy feedstock for bioethanol products. Our long-term vision is to really determine the industrial feasibility for large-scale bioethanol production from Sargassum across the region, which in and of itself will be a challenge because we have to take into consideration the techno-economic analysis, life cycle assessment, supply chain development, and establishment of regional processing infrastructure capable of handling millions of tons of seaweed annually. So when we look at our value creation pathways, you realize that the industrial application of Sargassum itself is endless. So it can be used in bioethanol, in bioenergy, in terms of bioethanol and biofuel production, for transport and industrial sectors. It can be used in agriculture as fertilizers, biostimulants, and animal feed supplements from residual biomass. So just to answer our interested participant online who asked about what will we do with what is left behind, we're trying to create a model that's circular. So that means no waste is created. It can actually also be used in cosmetics. When we go through some of the chemicals that we've seen in the Sargassum, you'll see that it has properties in terms of anti-aging, antibacterial, and that can contribute to formulations for skincare products and skincare products. Most skincare products are actually derived from marine extracts. Pharmaceuticals in terms of the bioactive compounds for antiseptic, anti-cancer, and antiviral applications. So when we look at the chemical composition of Sargassum, so Ruth would have touched on this, the tetracosane, the pentacosane, the hexacosane, the octadecane, eugenol. I don't know if when you think about essential oils, they're all from the Caribbean. I'm sure you would have heard your grandparents talk about pimento oil or the oil from the clove. And there are other famous essential oils, right? That you always use when you feel sick for different ailments. Limonene, peppermint essential oil, lavender essential oil. All of those essential oils actually have eugenol, the turmeric, so like turmeric that we use in the Caribbean. Anytime we have breakouts on our skin, we always try to go for turmeric products. And limonene is actually something that's found in lemongrass essential oil as well. So the tetracosane is used in various industrial applications. So it is, because of its thermal stability, it can be used in biodiesel and petroleum production. So this is what the tetracosane powder looks like on the commercial markets, and it's sold for $50 US. So we can actually use Sargassum to produce this because we found that it is actually in the Sargassum at a very high quantity. Pentacosane, it is colorless, it's a waxy, long chain hydrocarbon, and it can actually be isolated from crude oil. It is actually used in petroleum again. So you'll find that most of what we found in Sargassum are things that can be used in petroleum production. But there are other very interesting compounds that can be used for natural waxes. When you look at the hexacosane, again, it's insoluble in water, so It's again used in thermal energy storage, but we'll get to the eugenol, which is used in food and beverages as a flavoring agent. It's used in medicine as well. So I spoke to you about the cloves. I know this is a very famous one that's used throughout the Caribbean to make essential oil, and that is because of the eugenol that's found in it. So it's rather groundbreaking that we found eugenol in Sargassum. Now, in terms of the safety of the application, because Sargassum actually has arsenic acid, which is something that we've been very cautious of, but we are exploring ways of extracting the eugenol and reducing the interaction of that with the arsenic acid as well. So eugenol, again, is commonly used to treat toothaches, dental fillings, and it's almost like a natural anesthetic product. And it can be used in cleaning products as well and agricultural applications like pesticides. Another project that we've been looking at in terms of using the sargassum because it has eugenol is, I don't know if you have, well, I do know because I've seen it at the hotel that we're staying at, the mosquito problem, right? So we're all in the Caribbean, we have mosquito problem. Eugenol can actually be used as that spray, right, as a repellent. So that is another possible application of the sargassum, a small scale application. So if you were into natural products, you could actually process the sargassum, extract the eugenol, and produce insect repellents and sell online or sell in your community or try to scale that at a commercial level. The turmeric has several uses as well, anti-inflammatory, and it's good for neurological disorders. But as I mentioned to you, as being a Caribbean girlie, it's something that we use on our skin. You know, we have our turmeric face mask. We use it a lot in Jamaica. I know there are nuances when it comes on to the Caribbean islands, but it's actually very good for anti-cancer. So you'll find that people like to drink turmeric tea, and it is because turmeric has the turmerone, which makes it good. Another thing that supports the sargassum for being such a good insect repellent is the fact that it has this compound, the turmerone, which is a natural insecticide and repellent in and of itself. So this one now is the limonene, which has, again, it can be used as a fragrance, it can be used to flavor things, it can be used in household cleaning products or as an industrial solvent. It is actually used in food, beverages, cosmetics, perfumes, and soaps. But I guess with the sargassum and with the arginic acid that's found in the sargassum, the applications might not be as open to the food industry and beverages. So you'll realize that in Ruth's presentation, she didn't mention anything about food. And so I will not encourage anyone to actually try to ingest the sargassum, even though I'll share this with you because while being in the field, collecting the sargassum and interacting with people, fisher folk in Jamaica, they have actually reported to us that when they collect the sargassum, they will take it home to their families, and the children who are troubled with asthma, they will boil it for them to drink as tea, and it seems to have some respiratory ailments in terms of helping with the asthma, even though there is no study to that, but people in different coastal communities have been using the sargassum in different ways already. Because of course, before you start to do the research, people will try to explore on their own to see. So we have not had or heard of anybody dying from drinking this tea that they've been making. but it's not something that we can support at this time, but the university is committed to doing further research to find out if it's something that we can encourage, or if there is any truth to the sargassum actually helping with respiratory ailments. So we spoke about the fact that sargassum is pretty good with bioenergy. We spoke about that. With agriculture, because of, again, it's really the residual application when it comes on to agriculture. So after you've used the sargassum to produce your cosmetic or your bioethanol, then the remaining residue, depending on if it's rich in potassium, calcium, and magnesium, this can actually be used or as a organic fertilizer, creating a secondary revenue stream for those small businesses who might be interested in collecting that large compost sargassum and then producing or making a product. It can be used as a natural biostimulant as well, which contains growth-promoting compounds such as alginate and mannitol, which stimulates seed germination and enhance plant vigor. So these bioactive molecules trigger beneficial plant responses beyond simple nutrition. It can also be used as an animal feed supplement, and having done some research before coming to the Bahamas, we found that there was actually a group back in 2023 here in the Bahamas where they looked at using sargassum as a biofertilizer and as an animal feed supplement. And I know that there's a company in Jamaica that actually processes the sargassum into pellets for goats, so they use it to feed livestock. For the pharmaceutical and cosmetic applications, because of its anti-inflammatory and antioxidant compounds and antiviral activity, it's perfect for cosmetics and pharmaceuticals. It demonstrates very exceptional potential as an antiseptic, anti-cancer, and antiviral drug. Again, the challenge is just really to isolate these active compounds and actually put them to good use. There are quite a lot of economic and policy impacts when it comes on to the application of sargassum. Quite a lot of governments across the region have been incentivizing industries for going green. And so as a result, if you were to engage in renewable energy production, utilizing sargassum, you ultimately cut your carbon footprint, which could lead to you being more market competitive because the government will give you some amount of incentive for reducing your emissions. So financial incentives for being more ESG focused as an investor, and you can also get green financing in terms of those kinds of opportunities coming your way. For regional energy security, the reason why we are encouraging countries and other research collaborators to come on board with us with this project is when we encourage you to create your own bioethanol or have your own bioethanol production industry, it reduces your dependence on external partners. And we do know that it is a very imbalanced world, not so stable in terms of energy and importing and the ethanol. If there is war, if there is any kind of disaster, then prices usually increase. And so as a result, it is an economic opportunity for us here in the region to develop a viable industry from something that seems to be affecting us alone, right? It's affecting mostly Caribbean countries. And so it presents an opportunity for us to produce our bioethanol here in the region and reduce our dependence on exporting. Beyond the industrial value chain, I mean, there are so many opportunities because Sargassum itself, in terms of the massive accumulation of Sargassum, it creates quite a lot of challenges for the environment. And so when it is that we are encouraging the potential use and application, we are contributing to climate action, we are reducing the amount of carbon dioxide that is in the atmosphere, we are also reducing the amount of carbon dioxide that feeds into ocean acidification. This process also actively mitigates climate change through sequestration of atmospheric carbon dioxide into the soils and roots of biomass. And there's one thing to consider: the carbon benefits we found that it's more beneficial when you do the aquaculture in terms of the seaweed aquaculture versus the actual the actual collection of the sargassum off the coast. So there are also opportunities for cultivating the sargassum near coast, and there are benefits to that. There are nutrition removal, it has a high nutrition removal capacity in terms of it has been linked to significant reduction in nitrogen level, and may also be used as a tool for the reduction of heavy metal concentrations in coastal water. And this speaks to the bioremediation capacity of the seaweed itself. There are public health considerations with sargassum, as mentioned before. When it accumulates along the coastline, it creates hydrogen, sulfide, and ammonia, which can lead to respiratory irritation, and it can affect people's skin. And it has been linked to adverse neurocognitive effects and headaches, nausea. And so it is a problem that it is of utmost benefit to us to manage, especially to collect the picture that Ruth would have shown. It's something that we want to manage. It's not something you want to have just sit there. But instead of just collecting the sargassum and burying it, as we've now come to learn that that's what they've been doing along Cable Beach in the Bahamas, we can actually use it to produce bioenergy, cosmetics, or pharmaceuticals. And so it is very viable for us to explore all of these opportunities. It doesn't have to be bioethanol, it can be a product of your own using any of the compounds that we found. All you have to do is pick one, isolate, and you can work and develop whatever it is that your heart tells you. So thank you. So at this time, we will have a presentation by...
Before
the presentation, we'll have questions.
This slide contains the solution to her problem. The one of seasonality.
With the cultivation?
Precisely. Precisely. In fact, if you were to cultivate on a large enough scale, you don't need to rely now on watching when they're going to come in anymore.
There are quite a lot of solutions to seasonality, like, for example, storage as well, which we encourage in terms of when you collect a large volume of sargassum, once you blend it and put it in the fridge or freezer, it can be stored for a very long time for future use. Seaweed cultivation is also potential use as well, because it does have a good enough replication time. So yes, I agree.
See, then you get to add that this is now a carbon sequestration project, say specifically, and apply for carbon credits as well.
Yes, you can apply for carbon credits. So that's what we were saying about the climate change opportunities, especially for industries and businesses. So if you go green, if you start producing your own energy, from Sargassum. It's something that your country, depending on the country that you operate in, they have incentives for that. Outside of that, you can apply for green bond, you can apply for grants through the Green Climate Fund just because you're doing something that supports the environment.
If you don't, let's say, do the farming itself, right? You could capture the seaweed that is coming in and hold that in holding pens so that you can now let it out as you need it, rather than relying on refrigeration. Because that's going to cost you money too.
Yes.
If you keep it in the natural environment, it'll keep doing what it's doing, earn the carbon credits, do the carbon sequestration, and you only extract it when you need it.
Mm-hmm. Okay, that's a very good idea. Thank you so much.
Dr. Bethel,
Yes.
if I may add to that. So the sargassum doesn't live very long. I think it has high turnover. So when you keep it in a pen like that, it tends to die quickly. It's harder to keep alive as you might think. Much harder than you think.
That's a good point. I agree with that, but then there is also benthic sargassum that that could be cultivated because it grows, and there's a beach with a lot of sargassum just growing close to the coast. So
when she says in a pen, does she mean like to take it from the ocean, then put it into a pen on land, or to keep it in the ocean? Because I'm talking about to keep it in the ocean.
Yeah, I know what you're saying, but I think it's more of a challenge. It's not so easy. It's not like, oh, just like contain it and it'll grow. And it's not a very easy to grow algae as compared to like some of the other ones that have already been put into aquaculture or mariculture purposes. The sargassum just doesn't do that well for whatever reason. It's quite finicky.
I agree, because if you should grow it, you wouldn't be able to grow it in a cage similar to the setup off the coast with similar aquaculture setups. It would have to be grown where it embeds itself along the coast, and you probably just
you can make a structure, like to give it something to look onto.
And there is enough of it. Like I don't think we need to go that route. Like there's an excess of it that
I have
yeah.
Sorry for cutting who was speaking. I just have a quick input. So I have a quick input. So with so there could be like what happened with most environment. There's always going to be site differences in a sense, and so by capturing a particular population or of algae at a fixed location could present problem in itself. Because what can happen at that location, if you have a lot of individuals in there, they're going to be competing for the same type of resources for survival. And if they're not able to kind of expand beyond that fixed point, then you can have issues with depleted resources at that fixed location. And on top of that, you can also have that site variability at that specific location to begin with. It would have already been depleted of maybe natural resources because of other organisms living in the area and using whatever resources that are actually available within that area. And so maybe having it just being in its natural state in terms of being able to freely float wherever it can float is could be the issues why they, when held in captivity, tend to die. And then again, you also have a variation in the ocean temperature, which I think shouldn't impact it that much because you do have the water coming in and out. But you just have to bear in mind that you have variation at sites. And that's one of the reasons when you do these environmental studies that involve sites, you need to account for that site differences, because if you don't account for that site differences in your analysis, you would be missing out on that important information that actually occurred at the site location.
Thank you so much for that input. I certainly agree with you as a scientist. Agree. And it's something for us to take into consideration, because when we had the same workshop in Antigua, the concerns were surrounding the seasonality of the sargassum. So I guess I'm trying to figure out how to cultivate it in a way that would be successful is something that would actually, it would be of interest to other parties in the region as well.
You can capture seasonality using mixed effects model, accounting for random intercept and slope, because what the random intercept slope does is that with the random intercept, you take into account the cluster, the main population of the specific site. So that would be like a cluster of sargassum, maybe from site A versus site B, C. And what the random slope does is that it assigns an intercept where each individual's varies, and so you can capture that seasonality. So a random intercept with slope is very, very good in terms of capturing seasonality when you're dealing with like statistical models. So it requires some of the more advanced stuff like mixed effects models, etc.
Ocean dynamics is gonna throw a spanner into that works right there. Yeah, because it isn't so much like knowing where they are, right? But it's the ocean dynamics that tells you how much there will be to begin with, and then where those will move. So typically, your ocean models will give you, you can, let's say maybe a week out, 2 weeks out, maybe tops. But anything beyond that is going to be very difficult, very, very difficult to predict. And so he's not wrong, but the ocean dynamics is going to make it a lot more complicated.
But you can account for spatial variation using generalized additive mix models, because those takes into account the non-linear trend.
Yes.
The non-linear trend versus linear. So you could count for both non-linear and linear trend now that we have this extra variable on top that introduces a non-linear pattern, because you have this variation where things are just distributed randomly all over the place rather than a sequence of a trend. You can then now capture the non-linear trend and the linear trend as well using generalized additive mix effects model, accounting for that spatial variation. So and it's understandable that you're going to have this variation with the anything you're dealing with water and ocean.
Zico, what is your background?
That's McKinsey. See, this McKinsey. I know McKinsey.
Oh, Dr. Bethel?
Yeah.
Oh, okay, yeah, okay, yes.
So we're on the same page.
Yeah, I know. I'm just trying to support what you're saying, and also letting know that there's.
GIS. Okay.
Yeah, I didn't recognize the voice.
Okay.
Thank you so much for that, Zico. We do appreciate all that you've said, as complicated as it is for our biological brain.
Well, I'm usually more good with the statistical stuff, so I have an understanding of complex models and non-linear linear trends. I'm actually working on some of it with my work that I've done.
Wow, this is really interesting. Thank you so much. So we solved the issue. We're halfway there, Ruth, with solving the whole seasonality issue. I think that would be groundbreaking for us. At this time, as we prepare to close, we do have a message from Taylor Cargill, a conservation scientist. She will discuss the Biodiversity Beyond National Jurisdictions Treaty, which is a UN Law of the Sea treaty that actually will come into force on January 17, 2026. And her presentation looks at the BBNJ and Sargassum opportunities for the Bahamas. I think it's an excellent note to end on, as she will also look at putting the Sargassum opportunities into the bigger international picture. What's international law saying, and how can we tap into those opportunities from the Bahamas perspective?
Good afternoon, everyone. My name is Taylor Cargill. I am a Bahamian conservation scientist currently calling in from the Cape Eleuthera Institute in South Eleuthera. So today I'm going to be sharing briefly about the BBNJ agreement, or the Biodiversity Beyond National Jurisdiction Agreement, which is a recent treaty that has emerged from the United Nations, and how it relates to sargassum, opportunities for innovation in the Bahamas and the wider Caribbean. So about my background, so my day-to-day work is mainly focused on conservation science and coral restoration, which means that I spend a lot of time thinking about how we can rebuild and protect fragile marine ecosystems. But another important part of my path has been involved in youth ocean advocacy. So this year at the United Nations Ocean Conference in France, I had the opportunity to be directly involved in advocacy around the BB&J Treaty or the High Seas Treaty. And being a part of that process allowed me to observe the behind the scenes part of negotiations, as well as to sit in on early implementation discussions around this treaty and the kinds of impact it can have on our region and on our nation. And this experience gave me a really deep appreciation of how global governance decisions can translate into regional and local action. So today I wanted to take the opportunity to bring these worlds together, the scientific lens and the policy lens, to explore how the Sargassum Challenge actually poses opportunities for us as a region and as a country to intersect with the emerging BB&J landscape. So first of all, about the challenge and how sargassum impacts the region. So across the Caribbean, we've been experiencing extremely persistent and disruptive environmental issue as a result of this influx of pelagic sargassum, which has been occurring in the Caribbean since 2011. And it's been having various impacts on our marine ecosystems, like our coral reefs and our seagrass beds, but also on human health because of the accumulation on our shorelines of sargassum can release toxic gases if it's not dealt with properly. But also on our fisheries and on our tourism economy, because of course we rely a lot on the cleanliness and the health of our shorelines for the tourism industry. So having this influx of sargassum isn't great for that. So this is a regional scale challenge, not just being faced throughout the Caribbean, but also to some extent in the Bahamas. And it's been driven by global ocean dynamics. So I have a map here showing the movement of sargassum throughout our shared ocean. And you can see two distinct patches. There's the Sargasso Sea and the Great Atlantic Sargassum Belt. And the Sargassum Sea isn't actually the problem. It's a natural, healthy ecosystem that supports a lot of marine life in the Central Atlantic. And it's just natural growth of sargassum that is kind of kept in that central area by the flow of different currents. But the problem comes from this Great Atlantic Sargassum Belt, which is kind of an anomaly or an offshoot of the main Sargassum Sea that emerged in 2011 and has been recurring ever since. And it's been driven by various shifts in ocean currents, but also in warming ocean temperatures, as well as in like pollutants and other nutrient dynamics in the Atlantic Ocean. So it's been overwhelmingly accumulating on our shorelines, but you can tell that it's not just a regional scale origin of the problem. It's actually a much larger scale beyond national waters, which means that it requires interventions that go beyond the local scale and incorporate regional and global collaboration to address this challenge. So some opportunities and the current momentum in the sargassum management area. Basically, regional scientific leadership has been aiming to turn the challenge of sargassum into an opportunity by using different strategies to innovate both for coastal resilience and for boosting our economy. So some efforts that have already been undertaken and are underway currently include forecasting and monitoring, for example, satellite models and marine spatial planning to try and understand the movement of sargassum through our shared ocean and through the ocean currents so that we can know how much sargassum is flowing into our region each year, and also how we can best harvest and dispose and manage the influx. Secondly, the sargassum bioeconomy, which is really exciting. It's an area of innovation where people are taking sargassum and finding creative, innovative ways to make use of it as a product, while also addressing the ecological challenge it poses. So this has been done and is being currently done in agriculture, biofuels, clothing and construction, pharmaceuticals, tourism products, and the list goes on. Thirdly, we have regional management partnerships that are being built and established around protecting and managing sargassum as a resource. So this includes the Sargasso Sea Commission, which the Bahamas is a signatory of, but it also includes work by the University of the West Indies in their Sargassum innovation projects, which I'm sure will be talked about later on, and also work throughout the CARICOM entity around sargassum. So on to the BB&J agreement, also called the High Seas Treaty. What is this? So this is a landmark UN treaty that was recently ratified this year in September 19th, and it enters into force next year on January 17th, 2026. This is essentially a treaty focused on conserving and sustainably managing marine biodiversity in the high seas. And the high seas are the areas of our ocean that are beyond national jurisdiction. So no one country, no one nation is responsible for caretaking of the shared ocean. It's something that has to be managed collectively through international cooperation. And this treaty is essentially striving to do just that. And the Bahamas ratified the BB&J Treaty Agreement in June 9th this year at the UN Ocean Conference. So we are a signatory and a ratifier of this treaty, and we have a stake in what this treaty looks like for our country and for the whole international collaboration. And this treaty actually has a lot of policy implications for innovation around sargassum. And it does this through 4 components. We have the marine genetic resources component and benefit sharing, which is designed to basically increase equity around small island nations and other countries like us having access to the benefits and the ability to participate in research on the high seas and gain from those genetic resources and the products that are being made from them. Secondly, area-based management tools like marine protected areas in the high seas. This creates opportunities for international collaboration for sargassum stewardship and stewardship of other ecosystems, but relevant specifically to this case because of protection of the Sargasso Sea and also management of the Great Atlantic Sargassum Belt, which is the problem area of sargassum flowing into our ecosystems. So it's a framework. This framework allows us to have better regional and global collaboration around these 2 key sargassum sources. Thirdly, environmental impact assessments. So the BB&J Treaty has protections in place so that we can have EIAs for activities in international waters, which are currently unprotected. So having this in place basically means that we have stronger oversight for activities that happen in the high seas, including industries that are heavily polluting and the potential impacts that this can have on sargassum proliferation in our region, because you can imagine increase in pollution and nutrients in the global shared ocean will have trickle-down effects on our region. So that's an important framework. And then lastly, the capacity building and marine technology transfer framework. This creates opportunities for us to be leaders and innovators and receive training and funding in developing sustainable sargassum solutions and innovating for value-added products. So basically, opportunities for training, opportunities for funding, opportunities for access to technology. And that's super important. So just a final overview of these kinds of opportunities, it creates a pathway for the Bahamas and for the Caribbean to be a leader in sargassum management through scientific cooperation for research and monitoring of sargassum, also through capacity building and finance mechanisms for Bahamian leadership in sargassum blue economy, also for technology transfer and benefit sharing from those marine genetic resources, which includes the sargassum, and then better environmental protections so that we can prevent expansion of the sargassum belts as a threat to our shorelines. So overall, we're seeing that sargassum, even though it's creating a major threat to the Caribbean region, it can also create opportunities for momentum and global frameworks like the BB&J agreement so that we can have opportunities to shift from a crisis response where we're just trying to manage and mitigate sargassum to a more proactive and science-driven response with long-term solutions that can support coastal resilience while also protecting and enhancing our economy. So for the Bahamas, we can align our national sargassum efforts with the BB&J agreement to open doors for research, for economy growth, and for youth leadership and stronger cooperation, not only across the Caribbean region, but across the globe. Thank you so much for listening, and I hope you guys have a great rest of your day.
Okay, thank you so much. At this time, I would like to say a huge thank you to the University of the Bahamas. I want to say thank you to my colleagues from the University of the West Indies. A huge thank you to our funders, the Foreign and Commonwealth Development Office. Thank you so much for attending, our lovely attendees who came in later, but nonetheless, I'm sure you were able to catch some of the presentations so far. Thanks a whole lot to our online participants who were greater in number, but you actually shared you asked the right questions, you gave us the insight that we came looking for and some information that we didn't know existed, but we definitely needed. So thank you so much. Yeah, have a great day and enjoy.
Thank you.
Thank you.
Keep in touch.
And thank you so much for the technical support. We needed the connection and the support, as you can see.
But do you think we were dummies down here?
No, nothing that at all.
Um, Thank you. One main charges. I don't recognize the faces.